Okay, so talking about this process of transcription, I really like this part of biology. I love how our cells code for genes and the beautiful technique that they use for this. And it's just very elegant in the way that we do this, and it's very efficient. So I really love this part of biology and to teach it to you guys. So here we have, let me get my pointer. DNA. Remember that DNA is double-stranded. I'm going to focus on this strand over here where we have a strand, and then on the other side it's highlighted in yellow. I'm going to talk about that in a bit. And then we have its complement over here. Its complement in DNA, it's double-stranded, is going to have the exact same genetic information and coding for a gene as the other strand. Remember that they're anti-parallel. It's the same gene, so you could take either one of those strands and use it as a blueprint. You're going to get the same end result. So highlighted in yellow here is going to be a gene. It's going to encode for a trait, let's say a protein, that the cell wants to make. And so appreciate that it only takes one strand, a part of that just one strand. It doesn't need both strands. So it takes that one strand and it's going to go ahead and say, okay, transcription is going to occur right there. I'm going to go ahead and start that process in order to end up with the protein that I want. So recall that in transcription, we're going from DNA strand, colored in blue here, to RNA. This is called transcription. So I can actually see that here's my nucleotides in DNA, A, C, and so forth. And so because of base pairing rules, I am able to get its complement in RNA. But remember that in RNA, we replace T with U. So instead of adding T here, our cells know to add the enzymes and all the different other workers that are involved in transcription. They know to add U. So A to U, U, G, C with G, that doesn't change. Remember that there wasn't a difference there. So C with G, G with C, and so forth. Just appreciate that there's no thymine here. Now we have our RNA complement. This is transcription, and this is the end product for transcription. So this occurs in 3 different steps. And I'm going to show you an animation that's going to kind of keep looping as I talk through this. I really like this animation. Just let you guys see that animation really quick before I begin talking. All right, step 1, initiation. RNA polymerase, that blue enzyme you see, that's kind of like a, gosh, I don't know what to, it's kind of like a candy corn looking shape, colored blue. It's going to attach to what is called a promoter. A promoter, highlighted in green here, a promoter is going to be the start of a gene. It's basically going to be a couple of nucleic acids that are already in your DNA. That's the promoter. It's already ingrained in your genetic material. And it says, hey, I'm the start of a gene that you probably want to code for. Start here. That's called a promoter. It's an area of our nucleic acids that's going to initiate RNA polymerase to attach there, start transcribing for a gene. Step 2, elongation. Now we see that RNA polymerase moving towards the right side of the screen. That guy is elongating, adding RNA nucleotides, kind of that red line that we see in the 5 to 3 direction, just like in DNA replication, that RNA polymerase is adding RNA nucleotides, elongating that strand. Step 3, termination. That RNA polymerase reaches what's called a terminator. Terminator is very similar to a promoter in an idea where it's a certain area ingrained in our DNA that's going to signal the end point of a gene. We can appreciate that RNA polymerase, it reaches the terminator. It will stop the elongation process. We have our end product, that single-stranded RNA strand that will go on. Okay, let me let you guys finish seeing that, and I'll move on. So this is everything I just talked about. If you would like to see it in written format, kind of bullet point, there you go. Pause the video if you would like to read it even further. This is another image showing you guys transcription. Kind of like a standstill image if you don't like looking at that animation. There's our promoter DNA already in the genetic material. RNA polymerase knows that that's the start of a gene and it wants to attach to this area to begin elongation. That is step 1, initiation. Step 2, elongation. RNA polymerase then lays out RNA nucleotides. You can kind of see that red line here. RNA polymerase is polymerizing that strand, elongating it. Step 3, by this point, RNA polymerase has reached what is known as the terminator, that area highlighted in red here, the DNA that tells that RNA polymerase, hey, stop here, the gene has ended. And termination then signals for that RNA polymerase to let go, stop what it's doing, and we have now our completed RNA. This is called transcription. We have gone successfully from DNA to RNA.